Hello, my name is Chamara Gunaratne, and I have been studying acute myeloid leukemia, or AML. AML is a blood cancer that frequently features a mutation in the epigenetic regulatory gene DNA methyltransferase 3A, or DNMT3A for short. This protein is known to regulate gene expression by turning on or off genes by methylating DNA. Patients who have mutations in DNMT3A have a higher chance of relapse and a more severe prognosis. My project is focused on how the mutations in DNMT3A affect its protein interactions within the cell. This is important because these protein interactions can impact how DNMT3A functions within the cell. Currently, I have developed the tools necessary to investigate this question. In order to determine which proteins are associating with DNMT3A, we decided to do a co-immunoprecipitation reaction, where an antibody would bind DNMT3A and we would be able to pull the antibody along with DNMT3A and any of its current binding partners. However, DNMT3A does not have a highly specific antibody, so we decided to clone a hemagglutinin tag to the end of the gene. This way, when the gene was translated in the cell, this HA tag would be attached to the end of it. If you take a look at the upper top middle portion of my poster, you can see the overall strategy that I used. I cut part of DNMT3A out of this plasmid using restriction enzymes and then used PCR to add the HA sequence. This new fragment was religated back into the plasmid and the sequence was verified using DNA sequencing. From there, I needed to transduce the DNMT3A HA plasmids into the mammalian 32D cell line. However, we had several different viral packaging systems, so I tested each one using a GFP-containing plasmid, since it would be easy to determine which was the most effective at transduction by simply viewing their fluorescence. When producing the virus in the HEK293 T cells, the RevGag packaging system showed the greatest fluorescence. However, after using the virus to transduce HEK293 T cells, Jurkat cells, and my target 32D cells, the Psi ecosystem showed the highest fluorescence. I determined that the Psi ecosystem would be the most effective for my transduction. My future goals for this project include transducing the DNMT3A HA plasmids into the 32D cells, verifying the proper protein expression using Western blots, performing the co-immunoprecipitation reactions to extract DNMT3A and its protein binding partners, and analyzing the proteins using tandem mass spectrometry.